want to say a thank you to CDHF and to Richard Fedork for organizing this. I think this is an incredibly important topic, and I think we'll look back in 50 years, some of us will, and I look at this era as a real tipping point in our understanding of the pathogenesis of human disease and our understanding of how we predict outcomes in individuals and how we treat disease as well. So I think this is incredibly important. But I think the other theme here is that we're really at the early stages of understanding the microbiome. And I think from some of the questions, there's a real hunger for clear answers. And there often aren't clear answers and sometimes more questions. I'm going to speak a little bit about health and disease, but we have to always start with the cocktail party trivia, and we've already had some of this today. We know that we're outnumbered by our bacteria by a number of 2 to 1. And if you look at the number of species of bacteria living in each of your colons, it's at the low end estimates about 5,000. Some would estimate as many as 35,000 species of bacteria that reside within us. And only a fraction of those have been characterized in any meaningful way. Your mouth has between 300 and 500 species of bacteria, so think about that. And your skin is crawling with about 120. But of all those species, only about 100 are actually pathogens. So the vast majority of the bacteria we live with are either friendly to us or we just really haven't characterized them. So I think it's changing really our view of the microbiota. I think traditionally we've thought of the bacteria in our gut as a foe, and the lining of our gut something of a fortress that protects us from the evil influence of the bacteria in the gut. I think we've had a complete revolution in understanding, and now we think of these gut bacteria as our foes, our allies, something we embrace, something we interact with, and ultimately something perhaps we can use to our advantage in maintaining human health. So, complex slide just as an illustration. We've talked about the microbiota and health, and I think Richard in his introduction alluded to this balance, and it's truly a balance. And this is a figure from a recent paper that looks at all the interactions, some of the interactions, so it's a very small slice of the interactions between bacteria in our gut and us as hosts to maintain a lot of normal physiologic functions of human beings. So in terms of maintaining our immune system, what we call immune competence, maintaining this tolerance so that some things we let go, we don't attack everything in our gut, maintaining the integrity of the lining of the gut surface, some aspects of our nutrition, obviously of interest to the majority of the audience here. Some effects also on things like drug metabolism. That's a very underexplored area, how the bacteria in our gut influence our absorption, response, metabolism of medications, and arguably even our behavior. So all these things are regulated to some extent by gut bacteria. So disturbing gut bacteria can obviously influence many of these things individually. And this is another figure from a recent publication that points to some of the human conditions where disturbance in gut microbiota has been implicated as a step in their pathogenesis. So you can go through this long list of things. We've already touched on obesity. There may be a role for gut bacteria in fatty liver disease, perhaps related to obesity, multiple sclerosis even, chronic fatigue syndrome, coronary artery disease, platelet problems, this ITP that you've commonly heard discussed. Again, type 2 diabetes mellitus, all wrapped into this metabolic syndrome, and some gut disorders as well. C. difficile infection, which we'll talk about, irritable bowel syndrome, which I'll talk about, and Dr. Collins after me will discuss some more. And we've already heard about the potential role in inflammatory bowel disease. So these are some of the things which have been looked at. Our understanding of the role of gut microbiota in many of these is still quite early, quite immature, and I'm sure as years go on, we're going to add to this list of human conditions where the gut microbiota plays a key role in pathogenesis. So this can happen through a number of mechanisms, and I realize this might be a little hard to read at the back of the room. But if we change the gut bacteria, a lot of things can be altered. Of course, that change can happen for a number of reasons. We've talked about this, different genetics, changes in diet, in medication exposure. And some of that over time could shift your gut composition, or could in the early stages determine your lifelong gut composition. What can change? Well, the microbiota has an effect on something called intestinal permeability, which is the leakiness of the gut lining. So how well molecules, compounds within the lumen, within the inside of the gut, can actually travel across that wall and enter the deeper tissues of the gut and influence things like your immune response. So permeability can be altered. We can have changes, as you've already heard, in short-chain fatty acid production. And those short-chain fatty acids may have metabolic consequences. We can have something called molecular mimicry, where components of the bacteria can trigger an immune response targeting that bacteria. But the markers on the bacteria that trigger that response can overlap a little bit with human tissue, so it can actually inadvertently trigger an immune response to human tissue. These are these autoimmune disorders which we discussed. So it's possible the gut microbiota play a role in the development of autoimmune disease. And also, of course, if we have changes in our gut bacteria, it changes our defense mechanism against infectious processes, and that can lead to gut infection and to systemic infection as well. So these are just a few of the pathways by which disturbances in the gut microbes can lead to changes in human health. So we'll talk a little bit about post-infectious irritable bowel syndrome, and Des alluded to this in the Walkerton cohort, which I'll show you a bit of data from our study of the Walkerton cohort. But to set the stage, post-infectious IBS is something possibly many of you in the room have experienced. It's a persistent change in bowel function with abdominal discomfort that develop after an acute infection, an acute gastroenteritis. And even though you get rid of the bug that triggered the infection, you're left with this chronic disturbance in gut function that you didn't have before. So some of you may have traveled to some foreign climate, been perfectly healthy, you had a Montezuma's revenge, got rid of the bug, you came home, and things just haven't been quite right since. And this phenomenon, as we'll see, can go on for years. So we know what happened in Walkerton. This was, I think, a major public health disaster in the history of Canada. It was no one individual's fault, although some individuals have certainly shouldered part of the blame. There's a series of failures in our defense system against contamination of public water supply. And, you know, in truth, the good that's come out of the Walkerton has been greater attention to maintenance of public water supplies and their safety. But of course, this was a disaster. The town of Walkerton had its drinking water supply contaminated with E. coli 0157, as well as some other pathogens, including Campylobacter. And of course, people who drank the water supply in the town became desperately ill. And of course, you will remember it took some time to recognize what the source of that problem was, and hence, unfortunately, some disastrous outcomes for individuals. So what happened? Well, probably if you estimate, there were about 2,300 people who got sick in Walkerton at the time, and that's about half of the town's population. It's a town of about 5,000. A lot of people got hospitalized. Some people developed this severe kidney problem called hemolytic uremic syndrome, and 6 or 7 people, depending on how you count who died directly as a result of the illness, but 6 or 7 people died because of this infection, including a number of children. It was really a gross tragedy. Well, this was an unfortunate human experiment, seeing what happens when you introduce a severe infection to a arguably healthy population in the town. And with Steve Collins, who's going to speak next, we have done a number of studies of the population of Walkerton to see what happens. Because this is an example of people who are healthy, they get an acute insult in terms of an infectious process, and presumably that has some acute and chronic disturbances in the microbiome that could lead to chronic changes in human health. Certainly when we went to study the population 2 years after the outbreak, we already saw that the proportion of people who had symptoms of irritable bowel syndrome, this IBS-like picture, was much higher among the people of Walkerton who'd been exposed to the infection than among the people there who'd managed to stay well during the outbreak. So even 2 years later, there's a significantly higher rate of IBS among the Walkerton residents who'd had the infection. We actually followed that cohort out to 2008, which is 8 years after the outbreak in May of 2000. And even 8 years after the infection, you could see chronic changes in gut function, symptoms, changes in bowel habit, and discomfort that fit within this phenomenon we called IBS among the Walkerton residents. So even 8 years after the outbreak, there was this persistent change in gut function that we label as IBS, and in this case, post-infectious IBS. So really an example of how you can disturb the microbiota in some way with a gross insult and lead to chronic changes in human health. The question is really why this happens, and we did one study in Walkerton where we measured intestinal permeability, where you get people to drink a particular kind of sugar, and you measure what comes out in their urine, and you can kind of estimate what kinds of sugars managed to get through the lining of the gut, and then infer how leaky that gut was, and that's that measure of that phenomenon called permeability. And lo and behold, we did find that the people there who had IBS-like symptoms, in fact, had increased intestinal permeability. So we don't really know how they got there, but there's a really plausible argument that changes in gut flora, changes in the microbiota, could lead to this change in permeability. And we've actually done genetic analysis in the population too, and identified that some of the genes that put people at risk of developing post-infectious IBS in fact relate to something we call the innate immune response, which is that very early first-line response against bacteria in the gut. So people who have a bit of a different response to the bacteria get more IBS, and they seem to also develop this change in gut permeability. So it's a very tragic event in the history of Canadian public health, but we've actually, I think, learned something about the long-term consequences of acute infection in otherwise healthy people. So the question then comes in IBS, is there a change in microbiota that we can measure and that explains that change in symptoms that we call IBS in an otherwise healthy population outside the setting of Walkerton? So we call this garden variety or sporadic IBS in the general population. And there have been a number of studies trying to look at this, and I'll just point to a few as examples. This was a study which looked at a small number of IBS patients and control patients who were similar in age and gender but didn't have IBS. And it measured organic acid production. And Dr. Madsen's already alluded to changes in bacterial output that can influence human health, and it's a measure of the changes in the populations of these bacterial species. And sure enough, in this study, the patients who had IBS had different profiles of organic acid production, things like butyric acid, acetic acid, propionic acid. An interesting thing in this study was then they correlated those levels of acid production, organic acid production, these are short-chain fatty acids. Levels of those acids seems to correlate with the symptom severity among the IBS patients they studied. So there's some evidence that the products of these bacteria drive symptoms, not just the mix of bacteria themselves. Of course, science is progressing, and the technology we have at hand to measure these profiles of bacteria in the gut have evolved. And this is just an example of a recent study which has looked to profile the mix of bacteria in patients with IBS and compare them to the mix of bacteria in people who are matched healthy controls. This uses something called 16S ribosomal RNA sequencing. So it's a lot of medical jumble, but essentially the ribosomes, these little factories in your cells, the nucleus of the cells, that transcribe DNA into proteins. So that's where everything comes together to match the DNA up and make a protein. And it turns out that the DNA that makes those ribosomes themselves is unique to each bacterial strain. So in fact, you can go back and sequence stool, for example, and pull out these sequences of DNA that allow you to estimate within that stool in this mix of bacteria exactly which species are present. But I shouldn't say species. That's probably not the right word to use. Species is a very specific name of an individual bacterium. As you saw from Richard's slide, we actually go up several layers up to things like phyla, which are broad classifications of bacteria. And really, for the most part, these studies look at large classifications. There's a lot we have to get down to the individual level of individual bacteria to really understand at the granular level what changes have happened. But in a general way, you can say that patients with IBS in this study seem to have more what we call firmicutes, which is one broad classification of bacteria, and perhaps fewer Bacteroidetes, which is another phylum, another big classification of bacteria. Okay? But you can see these shifts that seem to correlate with IBS symptoms. Another similar study from last year, another cohort of patients with IBS compared to healthy controls. You can again do these fancy analyses and produce what's called these hierarchical trees that look at the distribution of bacteria in a group of people with IBS. But then you can kind of go a little further. You can try to pull out subgroups that seem to have similar patterns. And that's, I wouldn't worry what the axes are and what the graph on the right represents. But at the end of the day, you can start to see these patterns. So you can say, okay, I've got this pool of people with IBS. I can pull out a little group that seems to have this profile of bacteria. I can pull another group out that seems to have this pool of bacteria. Maybe start to divide this pool of IBS up into individual groups. Because I'll tell you, one of the big problems in working with something like IBS is that IBS is a condition, it's a diagnosis of exclusion, we call it. Right? So there's no test we do to prove that someone has IBS. It's really a process of excluding all of the competing diagnoses, and at the end of the day, people have this combination of diarrhea, perhaps, and abdominal discomfort that fits the pattern, so we call them IBS. But it makes sense that within that group of people that we label in the clinic as IBS, there are lots of different processes that lead to that common symptom presentation. And that group of people we call IBS is probably a group of dozens, hundreds, if not thousands of individual disturbances in physiology. And our challenge in 2013 is trying to divide people up into these subgroups and target therapy based on what's truly wrong. So this is an example of how understanding the microbiome might allow us to do this. We can take a group of people of IBS, maybe we can pull out little subgroups that have a particular disturbance in the gut flora, and maybe we can target treatment to alter that composition of gut flora and correct the symptoms in that individual. I think for one of the first times in medical science, we're to sit in a circumstance where our ability to generate data exceeds our ability to analyze data in many circumstances. And this is a good example. The technology for digging through stool, for example, and profiling which bacteria are there and drilling down not just to the phylum, but maybe in the future right down to the level of individual species and quantifying all this. Technology is advancing at rapid speed. What isn't developing so rapidly is the ability to analyze, synthesize, and dig through those data and try to find these sorts of patterns. So if anyone has children here looking for a career, I think bioinformatics, which is that statistical branch, is a great way to go, because we really are in need of tools that allow us to do that more efficiently. So does that give us an answer? Questions before, what can you use probiotics to treat IBS? This is an example of a trial where, yes, there was a positive result. This was a particular Bifidobacterium strain, which was given in 3 different doses to a group of patients who had IBS. It's an Irish study, so 362 primary care patients with IBS, treated with different doses of this probiotic strain. And lo and behold, the middle dose, which is that taller gray bar, actually had a significant benefit in treating IBS. And this is that bacterium strain that's now marketed as a line. So that bacterial strain actually has a randomized controlled trial supporting evidence in IBS, supporting its efficacy in IBS. And there's some other strains which have also been studied with clinical trials. But I will caution the number of probiotic strains which have actually been studied in any rigorous way for their impact on IBS is very few. And there's a lot more we need to understand. And I think the problem was alluded to early on by one of the other speakers, that it's a bit like saying pills are good for human beings, to say that probiotics are good for a particular condition such as IBS. What we're lacking is the ability to choose a particular pill for the human being with a particular problem, right? So we want to characterize not only what illness we're treating, but also the microbiotic profile of that individual with the condition in order to really match the probiotic that's going to have a response. So this is a very blunt way, just throwing one probiotic mixture or one probiotic strain at a group of people with a heterogeneous condition like IBS. And thankfully this one actually showed a signal, but I think there's a lot of work to be done in the future to match individual diagnoses and individual hosts to the right treatment. So I'll shift gears a little bit and talk about C. difficile, Pseudomembranous Colitis. So Pseudomembranous Colitis, as you know, is the medical condition which is caused by this bacterium called C. difficile, and which leads to all the health problems you read about in the media. And unless you've been hiding somewhere, you've probably heard of C. difficile, and certainly most of you will work around hospitals and healthcare facilities, and you know what a challenge it is to eradicate this organism and to treat the individuals who become ill as a result. So C. difficile causes this pseudomembranous colitis. This is a lovely endoscopic picture, and gastroenterologists love to show endoscopic pictures. But of what happens when an individual gets infected with C. difficile. These little white plaques you see in the lining of the colon are the pseudomembranes, and hence the name. We hate to see this because we know it can be a challenge to treat this condition and a challenge to prevent it from recurring. So C. difficile, if you're interested, is a gram-positive spore-forming bacterium. So that spore formation is part of the challenge. It kind of can hide in a little ball with a protective coating, and it can survive for a long time on surfaces in hospitals and healthcare facilities, and hence the challenge in hospitals for actually getting rid of C. difficile when they have an outbreak. It's toxin producing, so it's the toxins that produce that do a lot of the damage to the colon. There's fecal oral transmission, so you have to ingest these spores in order to become unwell. And of course, healthcare facilities are, unfortunately, the most common place of transmission. Having said that, between 5 and 15% of healthy people actually carry C. difficile, and it's an interesting subject in itself. They seem to live in some sort of symbiosis with C. difficile in the stool. Infants very commonly are colonized with C. difficile, and long-term care residents are often commonly colonized. So we heard about the problems with the elderly, loss of bacterial diversity, and risk for C. difficile infection. Well, if you're already colonized and you lose your bacterial diversity in a nursing home environment, for example, there's a very high risk of progressing to this medical condition of pseudomembranous colitis. So I'll just move on, and the pathogenesis of C. difficile at very reductionist levels is pretty straightforward. You have the steady state where you have a nice rich diversity of bacteria in the lumen of the gut. You have this controlled immune reaction sort of living in harmony with these bacteria. You get treated with antibiotics. The diversity in the gut gets reduced. The number and diversity of bacteria is lower. And if you then get exposed to C. difficile, that's a ripe environment for C. difficile to proliferate, grow, and cause injury. And you can show this. So there's a number of studies using this sort of similar technology, of this case pyrosequencing, looking at 16S RNA, DNA, what we've alluded to before, to show that people who get C. difficile have a reduced diversity, so they have fewer different strains of bacteria in their gut. And that's what partly what predisposes them to the colonization and to the consequences of that colonization. And this is an example of one of many studies which have demonstrated this. And you can show differences in fermentation profiles. We talked about this organic acid output. So people with C. difficile also have reduced output of butyric acid, for example. We've heard already of the beneficial effects of butyric acid in controlling inflammation and maintaining the integrity of the gut. And that production is reduced, and so that predisposes to injury and to this condition we call C. difficile or pseudomembranous colitis. So can we prevent it? Well, part of the traditional approach to C. difficile therapy is to give another course of antibiotics to get rid of the C. difficile itself. But if you think about that, you haven't actually addressed the underlying problem, which is that reduction in diversity, reduction in number of normal healthy flora in the gut. So plaguing treatment of C. difficile is this high rate of recurrence. Yes, you give antibiotic, you get rid of the C. difficile, but the underlying diversity remains reduced, and the C. difficile grows back, and you get this recurrence. It requires another round of antibiotics. So the other way is to think about replenishing the bacteria and maintaining that diversity in the number of species in the gut. And so this is an example, what we alluded to before, using a probiotic strain to prevent antibiotic-induced diarrhea, and specifically C. difficile associated diarrhea. This is done in a hospital in Shanghai, and this is the mixture of bacterial strains which is actually marketed as BioK. So they gave BioK to patients in the form of a randomized controlled trial, and found that BioK therapy was associated with a reduced risk both of antibiotic-associated diarrhea in general and of C. difficile infection specifically. So that's a preventive strategy at the time of antibiotic therapy. And that shows some efficacy. So what you were talking before in the question and answer period, this is an example of a probiotic that could be used in that setting to prevent C. difficile in people getting antibiotics. But of course, another way to do this is to give people fecal transplants. So, if you're trying to restore that bacterial diversity, you can give probiotics by mouth. That's one strain or two strains in that example. Why not give them the whole gamish of normal colonic bacteria all at once in the form of fecal transplant? So this is the idea depicted here, and I'll just move on. This was an article in the New York Times this summer. Some of you might have seen a story of someone who saved their friend's life with a fecal transplant. She acted as the donor, gave her stool. Her friend had had bad C. difficile infection. She survived, and thanks to this generous donation of stool. So just quickly, fecal transplantation has been around for a while, and in fact the first report in the human literature is about 50 years ago. I won't let you look at that picture for very long. But it's been around for a lot longer, and this goes back into the 4th century and the 16th century, and there are reports of Bedouins using liquid warm camel stool to treat dysentery in the desert. So you ever get stuck in the desert, that's what you should do. You can do this at home. This is a YouTube video you can look up to find how to do this in your kitchen. But at the end of the day, it does seem to work. And anecdotally, any of us who do this know that people who've got refractory C. difficile, you think nothing's going to work, they've been exposed to several courses of antibiotics trying to treat it. You get near miraculous results with fecal transplantation, somewhere in the order of 90%. And this, just briefly, is a randomized controlled trial that was published in the summer in the New England Journal showing efficacy from a duodenal infusion of donor feces. So you can think about that. But it does seem to work. A word of caution, though, this is a report from the summer, actually just from last month, of a norovirus infection that seemed to arise in a recipient of a fecal transplant. And it's not proven, but it's an example of, I think, a broader concern that we can do a lot of good with fecal transplants, but we need to be cognizant of perhaps some of the bad that we could do in infection, and perhaps conveying some of these other conditions that we listed that can arise from disturbances of the microbiome. So I'll just conclude by bringing you back to this slide, showing you the effects of an altered microbiome on the human species. And I think, again, this is the beginning of a revolution in our understanding. I think this is just touching the surface. I think in the coming years and decades, this exploration of the microbiome is going to revolutionize how we approach the pathogenesis of disease, how we approach disease prevention, and ultimately how we treat our patients as well. Thank you.